This is audio recording for visually impaired students. Fermentation. Fermentation is the chemical transformation of organic substances into simpler compounds by the action of enzymes, complex organic catalysts, which are produced by microorganisms such as molds, yeasts, or bacteria. Fermentation. Technology is the use of microorganisms to produce food, pharmaceuticals, and alcoholic beverages on a large scale industrial basis. The science of fermentation is called zymology. Principle of fermentation process. The basic principle involved in the industrial fermentation technology is that organisms are grown under suitable conditions by proving raw materials meeting all the necessary requirements such as carbon, nitrogen, salts, trace elements, and vitamins. The end products formed as a result of their metabolism during their lifespan are result into the media which are extracted for use by human beings and that have a high commercial value. Types based on respiration. Aerobic fermentation or aerobic glycolysis is a metabolic process by which cells metabolize sugars via fermentation in the presence of oxygen and occurs through the repression of normal respiratory metabolism. Aerobic fermentation means that oxygen is present. Wine, beer, acetic vinegar need oxygen in the primary or first stage of fermentation. When creating acetic vinegar, for example, exposing the surface of the vinegar to as much oxygen as possible creates a healthy, flavorful vinegar with the correct pH. Homofermentation. When only one type of product is formed. Heterofermentation. When more than one products are formed. Lactic acid fermentation. Lactic acid is formed from pyruvate produced in glycolysis. NAD positive is generated from NADH. Enzyme lactate dehydrogenase catalyzes this reaction. Lactobacillus bacteria prepare curd from milk by this type of fermentation. During intense exercise, when oxygen supply is inadequate, muscles derive energy by producing lactic acid, which gets accumulated in the cells, causing fatigue. Alcoholic fermentation. This is used in the industrial production of wine, beer, biofuel, etc. The end product is alcohol and CO2. Pyruvic acid breaks down into acetaldehyde and CO2 is released. In the next step, ethanol is formed from acetaldehyde. NAD positive is also formed from NADH, which is used in glycolysis. Yeast and some bacteria carry out this type of fermentation. Enzyme pyruvic acid decarboxylase and alcoholic dehydrogenase catalyze these reactions. Citric acid. Citric acid 2-hydroxypropane 1,2,3-tricarboxylic acid derive its name from the Latin word citrus with a molecular weight of 210.14 gram per mole, which contains 3 carboxylic functional groups with 3 different values of pKa 3.1, 4.7 and 6.4. Citric acid, a primary metabolic product, is the most important organic acid produced in large quantity and is extensively used in food and pharmaceutical industries. Citric acid is a carboxylic acid formed in the tricarboxylic acid cycle or Krebs cycle and is found in small quantities in all living organisms. Citric acid was first commercially produced in England in 1826 from lemons contains 7 to 9% citric acid until 1999. It is produced mainly by submerged fermentation using Aspergillus niger or Candida sp from different sources of carbohydrates such as molasses and starch-based media. Citric acid production. About 99% of world citric acid production occurs through microbial processes, which can be carried out using submerged or surface cultures. The 70% of anhydrous or monohydric form of citric acid is used in food and beverage industry as an acidifier or antioxidant to preserve or enhance the flavors and aromas of fruit juice, ice cream, and marmalades. 20% is used in the pharmaceutical industry as antioxidant to preserve vitamins, effervescent, pH corrector, blood preservative, or in the form of iron citrate as a source of iron for the body, as well as in tablets, ointments, and cosmetic preparations. In the chemical industry, which uses the remaining 10%, is employed as a foaming agent for the softening and treatment of textiles. A. Surface fermentation. Using rice bran or other starch or sugar-rich substrates have been used for surface fermentation by aspergillus niger or in liquid solution in flat aluminum or stainless steel pans. Niger can be used to produce citric acid despite the high content of trace metals in rice bran are used. The citric acid is extracted from the bran by leaching and is then precipitated from the resulting solution as calcium citrate. B. Submerged fermentation. It is a common practice where citric acid is made. The fermenter is made of acid resistant materials such as stainless steel. The carbohydrate sources are molasses, decatenized by anion exchange sucrose or glucose, MgSO4, 7H2 and KH2PO4 at about 1% and 0.05 to 2% respectively are added. The pH is never allowed higher than 3.5. Copper is used at up to 500 ppm as an antagonist of the enzyme aconitase, which requires iron. 1 to 5% of methanol, isopropanol, ethanol when added to fermentation containing unpurified materials increase the yield. The yield are reduced in media with purified materials. As high aeration is deleterious of citric acid production, mechanical agitation is not necessary and air may be bubbled through. Antifoam is added. The fungus occurs as a uniform dispersal of pellets in the medium. The fermentation lasts for 5 to 14 days. Thanks for watching.